Okay, so the recording is in progress now. I'm Kyle. I'm the host of Planet Microbes. My pronouns are he, him. So my background is in biological oceanography. I have a PhD in studying how phytoplankton interact with the chemical environment in the ocean. Um, since then, I have left the lab and I'm now an editor at one of the nature journals where I handle manuscripts at the intersection of earth science and biology. So I spend all day reading papers like this and helping them through the publication process. And today at Planet Microbes, I get to share some of the cool stories that I come across. So I'm thrilled to be here and to talk about how microbes get cozy under the crater. So first, a little bit of background. We have to go way back in time to the Mesozoic, which was an era of Earth between around 250 million years ago up until around 65 million years ago. So things were, as you can imagine, pretty different on Earth back then. We can see that the configuration of the continents on Earth at this time through the Triassic, the Jurassic, and the Cretaceous, which are the three sub-periods in the Mesozoic, had a completely different formation than they do today. And of course, this is the period of time that is known for having the dinosaurs. And we all know what happened to the poor dinosaurs. They are gone today. And this occurred as a result of the end Cretaceous Chichulube impact. If I'm pronouncing that wrong, I don't think I've ever heard that pronounced out loud until it just came out of my mouth just now. So if anyone knows how to pronounce that, please hop on to the chat and say how that should be pronounced. But the important thing to know is that this was an enormous asteroid impact. The meteor was, when it came into Earth's atmosphere, was 6.2 miles across. And that meteor struck in the Gulf of what would today be the Gulf of Mexico. And as a result of that impact, it created this enormous ripple effect that basically led to a mass extinction, killed off all of the non-avian dinosaurs and about 75% of all life on Earth. And this was such a massive impact that there were debris, shrapnel that flew up from the impact site and spread thousands and thousands of kilometers away, as you can see in these little yellow dots. So scientists have gone out and sampled the geological record, digging through these sites that are really far away, and have found rocks that would only have existed right where that meteor struck in the Gulf of Mexico. This event also would have created gale or hurricane force winds that basically decimated all of the forests around the area. And it also would have created massive tidal waves that would have been felt at every coastline around the world. So this was a big deal, especially if you were a dinosaur. And this also caused major climate impacts around the globe. So first and foremost, all of that dust that was stirred up by the impact went into the atmosphere. That reflected sunlight, so plants had a harder time growing. This material in the atmosphere also caused acid rain to occur. So chemical reactions happened with all of that stuff that was in the atmosphere, which created acidic compounds that rained down. It also caused wildfires in the immediate aftermath of the impact. Dust and aerosols, which I mentioned, poked a hole in the ozone layer and led to other greenhouse gas production. So this was not a good time for everyone who was on Earth at this time. And you can see this impact in the fossil record. So here is a region that has had all of the material that has been deposited since this mass extinction event, blown away by erosion. And you can see this stark contrast in the fossil layer that's exposed from before and after that meteor hit Earth. And this event really demarcated the end of the Mesozoic, and then we entered into the period that is known as the Cenozoic. So this is a time where Earth started to look more like it is today, both physically. That's when the continents sort of shifted into their present configuration. The continents were where they should be. The connection between North and South America was bridged, and Central America formed, which separated the Atlantic and the Pacific Oceans. And this is when sort of life as we know it started to evolve into what we see in present times. And so the Cenozoic has lasted from basically right when that the Chichulube impact happened up until around present times. So of course, we know that, you know, all of the dinosaurs are extinct as a result of this event, but not everything died. You know, 25% of life on Earth lasted, and what else persisted throughout this event? There's a little hint at the bottom of this image.
Any guesses? They're so
the topic of the paper. Come on. Yeah, so microbes have persisted throughout every mass extinction event. So microbes have been around for billions of years. They have been around since long before humans were on Earth, and they will likely persist long after we're gone. So this paper is all about looking at how this crater impact in the Gulf of Mexico 65 million years ago, how it created this sort of friendly environment for microbes to grow that wasn't there before, and created a unique environment that fosters a special microbial community. So sort of creating this homey environment where microbes could thrive out of this event that rewrote Earth's history and completely decimated all life that was there before. And this research was first inspired, we have to do a little bit more background before we hop into the paper, but does anyone have any questions or comments before we jump into the more nitty-gritty science of this paper? Feel free to pop on into the chat or just use your voice to ask a question. Seeing some thumbs up, so we can probably.
We got one question.
Oh, sorry. Yeah. Sure.
So you mentioned that the continents were closer together when the asteroid struck. Mm-hmm. Was it like the Pangea construction where everything was like mushed together? And is that why you saw such a wide blast radius on it? Or
so that's a good question. I actually don't know how the configuration, exactly how the configuration of the continents would have changed the effects of the effects that were like immediately felt on land. So this is what the continents looked like, the Cretaceous 65 million years ago when that when the impact happened. So it would have happened right around right here. They almost look like they are today. The main difference is that North America is still connected to Europe and South America hasn't collided with North America yet. So I'm not, maybe the damage would have been more and more species would have gone extinct if North and South America were connected. That's a good question. So they were almost like they are today, but not quite. But I don't know if anyone's done reconstructions of how the effects would have been greater or worse depending on the configuration of the continents.
Can I ask one other question?
Yeah, sure, James.
The 25% of species remaining, 75% of species that went extinct, do those numbers include microbial diversity? And if so, what are the 25% remaining? What's sort of the makeup of the species that survive?
Yeah, that's such a good question. So typically the only way to reconstruct mass extinctions through the past is to use the fossil record. So they will look at marine samples, things that are buried in the sediment below the seafloor, or in terrestrial samples, and look at how long certain types of certain species persist through the fossil record. And then when those disappear, when you're going chronologically through time, then you can say this organism is no longer seen anywhere, it's probably extinct. And you can reconstruct through the fossil record. I have a slide on this, but at the very end. So I'm gonna blow my punchline, but for the sake of science, I will. So this is a record that I got courtesy of Wikipedia, thanks Wikipedia, of all of the mass extinction events based on the fossil record. So this y-axis is the number of genera or different fossils, and then each of these triangles are where you can see a dip in the fossil record that corresponds to the big 5 mass extinctions, which are the yellow triangles. So really we're limited by what we can see, and microbes don't typically fossilize very well or very clearly. So, you know, one itty bitty little circle looks a lot like every other itty bitty little circle. So those are harder to distinguish. Although I have seen some researchers lately who are using really high precision chemical techniques to identify microbial fossils. So maybe at some point in the future, we'll be able to answer that question, but for now we're limited by the bigger critters and plants that would be preserved in the fossil record. Great question. And then Will has a question. Any major differences between the pre-impact microbes and those post-impact? For example, what aspects enabled them to survive, or maybe what things were mutated or evolved as a result of post-impact chemistry? That's a really good question that I don't know the answer to off the top of my head. So you can look at the preservation of different types of minerals, or the chemical composition of the sediment in the deep ocean, to sort of reconstruct what organisms were living in the surface ocean before and after these times. So you can see, you know, a wipeout of not specifically what the identity of the microbes were, or what genes they had, or what metabolic capabilities they had, but probably plants and photosynthetic organisms had a harder time growing before and after because of the stuff that was in the atmosphere that was blocking out the sun. But I don't know if we have exact evidence of what the physiology was like before and after from exact samples, for the reasons that were in the answer to the question that James asked. We just don't, things aren't preserved. DNA isn't preserved very well for millions of years after these events. So it's hard to get an exact answer. And Kendall asks, the figures are global or at the impact site? So all of the figures from this paper are based on the impact site. And then this is, these figures right here are samples that are taken from the same site, but some of the authors' earlier work that came out in 2020 that was just looking at the structure of the rocks beneath this impact site, which is a good segue, if there are no more questions, into sort of the inspiration for looking at this crater site and how it makes that cozy home for microbes. So previously, these researchers went out to the middle of, or the side of the Gulf of Mexico, right at this impact site. And so if you, even today, 66 million years later, you can still see in the topography of the land in the Gulf of Mexico, the impact of the crater. So there's the center of the crater where the meteor hit, and then you can still see sort of like a mound of sediment that is the blast radius, or the immediate impact site. And these authors drilled down into the sediment in what they call a borehole, and took a sample to look at what are the lasting effects of this impact on the minerals that exist beneath the surface. So what they found in these samples of rocks from beneath the peak ring are more porous and permeable niches. And so this sort of piqued their interest. And this is because they realized, what do microbes need to grow? So if you're a microbe living in rocks, what are all of the ingredients that you need to be able to survive in this pretty harsh and inhospitable environment? Any ideas? Well, you need an energy source. So some sort of chemical you can break down to get energy, and you need to either find organic carbon to eat, or you need to make your own. So either doing photosynthesis, which probably is not going to happen very much deep below the surface of the Earth, because there's no sun. So they'll need to get chemo-autotrophic type of lifestyle, because they won't have the sun to provide that energy. And they will need some source of the building blocks of life, the nutrients that are needed to grow. And what it looked like is that as a result of this impact, it created this porous, permeable rock where there wasn't porous or permeable rock before. And the idea was that fluids, which you can see here in this schematic image, enabled fluids from the subsurface to sort of flow through these permeable, porous veins that ran through this otherwise really dense rock. And what they thought after looking at these was, maybe this would be a really good home for microbes, where otherwise it should be like totally inhospitable for life. No energy, no water, obviously no sun. And so that is the topic of this follow-up paper from these authors, in looking at really answering that question of, is this a hospitable environment for microbes? And did this crater create this special environment that could foster unique life where there wasn't any before? So before we hop into what this paper did, are there any other questions? Oh, Kendall, I see you said moisture. I missed that in the chat. But yeah, that's exactly right. That's another thing that microbes need. They absolutely need water to grow. In a past Planet Microbes, we talked about microbes that can extract water from rocks to grow. So they might find water in unique places. All right, are there any other questions before we jump in?
One question I had, the boreholes they took, when they initially took those boreholes, were they expecting to find, did they make the boreholes mainly to ask these questions about microbial diversity, or it seemed like the boreholes might have been part of a larger project to study Chichulube?
Yeah, so my understanding is that this was probably like an interesting follow-up or a happy accident. I can't speak for the authors or what they had planned, but I think these boreholes were part of like a global sampling initiative where there are similar samples taken all over the world, and you can answer so many questions by looking at various aspects of these sediments. So I think this is super fascinating. So this is an example of not the borehole that was taken for this paper, but a similar one. And these things are basically like time capsules that enable researchers to go back in time and look at the chemistry or the fossils that were preserved in these samples. So, the surface is present time, and then as you go back and sediment is deposited on top of the older sediment, you can go further and further back in time. And the chemistry, reconstructing samples taken from all along these sediment cores, can tell you about how much CO2 was in the atmosphere at the time, in the atmosphere or the ocean at the time that it was deposited. So you can reconstruct millions of years of Earth's CO2 in the ocean or the atmosphere. You can look at the temperature of whatever was above this sediment over time. So there are these really powerful tools to reconstruct Earth's past climate. So this was probably a happy accident, would be my long-winded answer for that. So what they were looking for is this suvite. I also have no idea, oops, there's my cat. Have no idea how to pronounce this particular mineral, but this is a particular type of rock that is only formed after an impact event. So you can see a different example of what this mineral looks like. It's sort of rocks that are all mushed together, as you can imagine would happen when you are hit by a 6.2 mile across meteor that is hurtling towards the planet. So they took this sediment core from this site in the Gulf of Mexico, right where the meteor hit. And then what they did was analyze the microbes that are currently living there at this time. So one thing to point out is they're not assessing the microbes that, these aren't microbes that survived the impact. These are microbes that have, in the ensuing 66 million years, have found a nice comfy home in the aftermath of the impact. So it is highly unlikely that any microbes or anything at all would have survived at the exact site of this meteor hitting, but these are the microbes that found their way there after that impact event. So what they did was take samples from down this core. So here on the y-axis are the depth of the depth of the core sample. And then here they're looking at the number of microbes that they counted using microscopy. And what they noticed is here at this depth of 650 meters, that corresponds to the sediment in the timeline of the core of when the Chichulube struck. So this is that special mineral suvite, and they found that at this particular band, they found a very high concentration of microbes relative to the other places around the core. So down lower here is pre-impact, and then above is post-impact. So all of the sediment that was deposited after that impact event. Moving across here, they also found that the amount of DNA they were able to extract was highest at this region. The amount of organic carbon was super low until after the impact event. And this also corresponded to a region of increased porosity. So that particular structure of the mineral that they thought might have enabled water and nutrients to filter through and allow that microbial life to grow. And what they found is that these impact regions in the sediment core have these super abundant and diverse microbial communities that are unique to this impact site. So here, looking at this box and whisker plot, doing a bit of light statistics on these cell counts, all of the rocks that were before the impact of the Chichulube had lower cell counts relative to that particular rock that is found only from the impacted sediments. And again, lower number of microbes in the post-impact sedimentary rocks. And it's important, I think, to remember that these are still themselves, even post-impact, buried pretty deep in Earth's sediment. And looking at this Venn diagram, they also found a number of unique microbial taxa that were only found or endemic to these particular sites. So 604 of these microbial taxa that they found are only found in this region that is impacted by the impact. And does anyone know how they assessed taxonomic diversity? Did anyone pick that up from the paper?
I think they sequenced the ribosomal RNA from 16S ribosomes.
Exactly, yeah. So that is a technique where you can basically fingerprint any microbe by using the sequence of their ribosomes. So ribosomes are the protein-making factories in cells, and generally every species has a unique sequence. And so they took this small snippet of genome that codes for the ribosomal RNA, and then sequenced that and used it to figure out who was who. So really the only thing that they are able to say is what taxa are present, and not necessarily everything else in their genome. So only who's there, but not necessarily what they can do. So I see Beth has a question in the chat. Is the sedimentary rock fused at the point of impact, but increased in porosity at the fringes of the crater? Oh, that was Kamal. I have no idea. So the part that they sampled is sort of that ring, that domed ring that forms all around the actual site of the impact. I think you can see it in this image. So this is sort of like the topography of this impact site. So you can see here in this black line, it's the sort of that little mountain of sediment that's created as a result of the impact. So that's stuff that's sort of pushed over. So I don't know what it would be like in like the exact spot where the crater hit, because they sampled slightly further out, but still obviously really close to the impact site. So yeah, it's a good question, but I don't know. Kendall asks, how deep below the surface the samples reach? Let's see. So this is, these are taken at a depth of between 500 and 1300 meters below the surface of the sediment, so 1,300 meters down.
Is that also underneath water?
I do believe it is underneath water at this point. Yeah. Okay, so they had this evidence that that this particular structure and characteristics of these minerals and rocks that are beneath the impact are paving the way for these unique microbial communities. And they do a bit more analysis on the 16S results, and they look at are these communities really unique? Is this environment sort of selecting for a special community of microbes that is different from everywhere else? And that's what this correspondence analysis plot is showing. So each of these little dots is a different microbe, a different taxa, and the colors correspond to green being before the impact in the sediment, purple being post-impact, and then right at that sweet spot where that unique mineral is are these triangles in pink. And so because they saw these different samples clustering together, that was good indication that these microbial communities do have a unique different fingerprint of abundance of different organisms. And that's borne out looking at the specific species that they found. So each of these column graphs show the percent abundance of different species. Each color is a different phyla, class, or order of bacteria. And you can see, again, post-impact, right at that impact, and then pre-impact, going from left to right. And you can see that the abundance of different microbes is different. And this graph is showing that that is statistically significant. So indeed, this environment created by the impact is selecting for a unique community of microbes. And then they wanted to look at what conditions specifically are driving that community composition. So in addition to sampling the number of cells and the 16S to show who was there, they also looked at a number of environmental parameters. So they quantified the porosity, the chemistry, looking at sulfur and metals like manganese and iron. They also quantified the amount of total organic carbon, which is what TOC stands for, and the temperature. And then by drawing these lines on this principal component analysis, they're able to show which of these factors are most driving the community composition in these particular samples. And what they found is that organic carbon and sulfur are the main parameters that are causing microbial differences between impact and post-impact, which sort of makes sense considering that we saw that stark shift in that plot going from surface to depth of organic carbon. And they also found that the conditions that are there, higher concentrations of iron and manganese and heavy metals, are indicative of metabolically versatile bacteria who are able to use these metals and cycle nitrogen and sulfur under the anaerobic conditions that are there. Things are still pretty hot down there, so they also found associations with temperature driving this distribution. And ultimately, that porosity is key to survival. So the fact that this mineral has more of a permeable structure enables all of these compounds to move through and fuel microbial growth. And they do make some inferences about metabolic capabilities, but again, like we discussed before, some of the key limitations of their approach are that they really are only able to tell who is there from 16S. So you can look at other organisms who've had their genome sequenced and make inferences about what their entire metabolism might be, but ultimately those are just inferences because they only have the snapshot of who's there. So that's an overview of this paper. Does anyone have any questions or comments or things that they liked about the paper, or things that they didn't like about the paper, or questions that they thought were left unanswered?
I have two questions related to this slide.
Mhm.
I feel like I intuitively understand how something like an impact of an asteroid could create porosity. You know, a big shake up and shake things up and gets a little more porous. But how could an impact like that lead to a difference in something like organic carbon or these other factors? And then the other question is, in this, how do they control for depth? Could it be that this different community is just a community that forms at this particular depth, not necessarily because it is forming at this this sous vide section?
So your first question, I think it's related to the amount of stuff, the stuff of life that microbes need to grow it has to come from somewhere. So the deeper you get in the sediment, things get really hot really quickly. And so that is sort of your limit on life. But organic carbon has to be created, has to be made somewhere to fuel life, whether you're making it yourself or you're getting it from something else. Obviously, there's no photosynthesis down here, so something has to be making that carbon or recycling it and then delivering it to these microbes to use. So something about the porosity that enables, you know, things to sort of percolate through the rocks and be delivered to the microbes. That's kind of a vague answer, probably.
But you're saying impact increases porosity, and then porosity also has this effect on available organic carbon, which also has this effect on the composition of the community at that particular strata.
Yeah, it just seems like this sweet spot in Earth sediment that is, I mean, there were microbes throughout. It's not like there were no microbes below. They were just far fewer because things get hot and there is limited porosity and limited access to energy and nutrients to grow. But for and actually it's a good, I'm wondering if porosity is higher or lower above the, oh yeah, so porosity is, maybe this is the key. So porosity is lower above the impact or post-impact sediment as well. So yeah, it's this sweet spot that enables the delivery of these compounds. So it could be, you know, their ability to get metals and nutrients from below and organic carbon from above, and those are kind of meeting and converging at this sweet spot that's making a nicer place for microbes to grow. That would be my guess. And then your second question about controlling for whether whether there's like a sweet spot everywhere around the globe. So yeah, they don't really have a control sample from like outside the crater in this study. Maybe that's another limitation, because yeah, they would have to test cores at like in Europe or something, or anywhere they have them at that particular depth.
Or even not all across the world, but just, you know, a few miles away, off of the rim. Seems like it.
Oh yeah, that would be easier, huh? Than taking samples from around the world. But yeah, I mean, they do mention that they don't think this is contamination because things are so hot down here, and it's different species that they don't see anywhere else. So yeah, maybe a forthcoming paper will compare this. I mean, there are, this is another thing that we talked about at a different Planet Microbes, but in sediment cores around the world, there are these sweet spots, similar sweet spots, where there's like a perfect mix of nutrients and organics from above and other nutrients from below, and oxygen that does create sort of a similar sweet spot for microbes to grow, where it's not too hot, not too cold. It's like a Goldilocks condition within sediments. But whether that, obviously it's not as a result of a earth devastating impact anywhere but right here. So this is only, I think, one of two impact sites that they can still see on Earth. I think I read that. Someone correct me if I'm wrong. But so that's a unique, it's a unique site. Any other questions or comments or anything? So I already kind of said this, but what I think is super interesting about, more philosophically compelling about this paper, is the hardiness of microbes, and the idea that these that microbes can find a way to grow anywhere, even deep beneath the surface of the earth, in rocks that are impacted by a meteor that wiped out everything on Earth. And there have been 5 major mass extinction events before, but some scientists think that we are at the dawn of the 6th mass extinction. And, you know, microbes survived all of the previous mass extinctions, and so they will probably likely survive this one. And I just think it's super interesting that microbes find a way to survive even in the most inhospitable of environments. So maybe the future is microbial. So that brings us to the discussion questions, and we are a teeny tiny group, so I think we can all probably do it together.
Yeah, I'm going to go ahead and stop the recording here, and that way folks can turn their cameras on, get comfy, and we can chat.